Now continuing our way to the next agenda. I'm honored to invite our first keynote speaker, Dr. Mi-Hua Tao, the Research Fellow and CEO of the Biomedical Translation Research Center, Translational Medicine Division. Dr. Tao specialized in virus and cancer immunology and gene therapy. The topic of the speech Dr. Tao is going to give us today is application of human immune system mice for cancer immunotherapy. Let's welcome Dr. Tao.
It's a great pleasure to participate in this 2022 Avatar Medicine for Patient Forum. My name is Mi-Hua Tao. I'm a research fellow and CEO of Academia Sinica. Conventional anti-cancer therapies, such as chemotherapy and targeted therapy, act on cancer cells, but they often fail to completely eradicate cancer cells and result in tumor regrowth and therapy failure. On the other hand, the recently developed immunotherapy act on immune cells, and they induce durable clinical response in a subset of patients. For example, ipilimumab act on CTLA-4, and nivolumab and pembrolizumab act on PD-1 that result in about 10% and 20% overall survival. To further improve the long-term survival, a novel anti-cancer immunotherapeutic strategies are needed, and for that, a preclinical animal model that contains both cancer cells and human immune cells are needed. Tumor microenvironment contains many cell types in addition to cancer cells. There are anti-cancer cells, such as cytotoxic T cells and dendritic cells. There are also pro-cancer cells, such as tumor-associated macrophage and cancer-associated fibroblast. A good preclinical animal model for testing cancer immunotherapy should contain patients' tumors, the so-called PDX mice, and functional human immune cells, the so-called HIST mice. And the source of tumors can be patients' cancer tissues, or cancer cell lines, or circulating tumor cells. And the source of human immune cells can be CD34 plus hematopoietic stem cells, HSC in short, or peripheral blood mononuclear cells, PBMC in short. As mentioned in the previous slide, patients' cancer tissues or cells can be transplanted into immunodeficient mice to generate the first generation of PDX or CDX mice. These tumors can be further transplanted into secondary recipients to further expand the available tumors for different applications. These mice can be used for drug screening to select the most potent drug for treatment, or to rebuild tumor microenvironment, or to be used for research such as discovery of gene signatures in tumor cells. For example, the PDX mice from one patient are divided into several groups and treated with different therapeutic regimens. In this case, treatment B produced the best anti-tumor effect in the PDX mice, and thus is selected to treat this patient. This slide shows application of PDX mice to evaluate therapeutic efficacy of human natural killer cells. The mice were injected with luciferase expressing human ovarian cancer cells, which also express high level of a tumor-associated antigen called mesothelin. These mice were then irradiated and received injection of 15 million natural killer cells prepared from different sources, including peripheral blood, inducible pluripotent stem cells, or iPS that express mesothelin-specific CAR, IL-2 and IL-15 was supplemented to these mice to support growth of human NK cells in vivo. Before treatment, all groups show similar tumor size. Compared with the no-treatment group, treatment with peripheral blood NK cells or iPS NK cells showed significant anti-tumor activities. Expression of mesothelin-specific CAR further improved the anti-tumor activity of NK cells. This slide shows generation of human immune system mice using hematopoietic stem cells. Mice were first irradiated to deplete murine bone marrow HSC niche for human HSC. The source of CD34 plus HSC can come from umbilical cord blood, bone marrow, fetal liver, or GCSF mobilized peripheral blood. In early experiments, non-skid mice are used as hosts, but these mice usually gave low engraftment rate. IL-2 receptor common gamma chain deficient mice, such as NSG, NRG, and BRG mice, are commonly used now. These mice gave about 45% engraftment rate in 2 months. The major leukocyte populations are T cells, NK cells, and B cells. However, these immune cells usually gave poor immune responses. For example, when mice immunized with keyhole limpid hemocyanin and complete from adjuvant, they only induce low IgM antibody, and no IgG antibody can be found. T cells from these mice stimulated with PHA or anti-CD3, anti-CD28 antibody only induce low titers of T cell proliferation as indicated by the white bar as compared with T cells from adult blood, as shown by the black bar here. There are several methods to increase human immune cell frequency and functions in HSC mice. The first method is by co-expression of human HLA. In T cell development, progenitor T cells need to interact with thymus epithelial cells for MSC-restricted positive selections. However, in humanized mice, there is no human HLA present in mouse thymus to support maturation of human progenitor cells. Dragon mice are transgenic NRG mice co-expressing human HLA class 1 and class 2 molecules, which can support antigen-specific IgG antibody response and T cell response. The second approach is by cytokine supplementations. Cytokines are required for immune cell differentiation. For example, T cell differentiation requires IL-7, B cell requires BAFF, and macrophage require MCSF. Mixed TRG mice co-expressing TPO, IL-3, GM-CSF, MCSF, and SERPA, and this mouse produce good myeloid cell lineages, including macrophage and dendritic cells and NK cells. The third approach is by engraftment of human fetal thymus. BLT mice are engrafted with hematopoietic stem cells, plus human fetal thymus and liver tissues, and this mouse can produce robust antigen-specific immune responses. Next, I will present a recently published paper using human immune system mice to investigate the role of macrophage in cancer metastasis. Mixed TRG mice and ISTRG mice were used in this study. Mixed TRG mice express 5 human proteins as described in the previous slide. ISTRG mice express 4 of the 5 proteins, but not MCSF. Transplantation of CD34 HSC resulted in 60 to 80% in graft burn rate at week 16. The MSTRG mice, represented by the orange circle, ISTRG mice represented by the purple circle. Analysis of these cells revealed that both mice supported development of T cells and B cells, while only MSTRG mice produced much more myeloid cells and tissue macrophages. This result suggests that MCSF is required for development of human macrophage. These mice were then subcutaneously injected with a human melanoma cell line called ME275. The tumor growth rate was similar in both mice. Macrophage infiltration, which is labeled as red color, was only observed in MSTRG mice, but not ISTRG mice. Interestingly, there is more cancer metastasis in MSTRG mice, including that in liver, lung, and spleen, much less in ISTRG mice. These results suggest that macrophage support cancer metastasis, and this animal model will be useful for developing drug targeting tumor macrophage. Another method for making human immune system mice is by transplantation of human peripheral blood mononuclear cells. The first step is by injection of about 10 million PBMC to NSG mice. As shown in this slide, only PBMC transplanted group produce human CD45 cells, but mouse CD45 cells were present in both groups. About 90% in graft burn rate can be achieved at 2 to 3 weeks. Most of these human leukocyte cells are T cells, greater than 97% in different organs. However, all mice develop severe graft versus host disease, and mice started dying at week 4. This is a xenogenic response, as human T cells are activated by mouse MHC and attack host tissues. The PBMC mouse model has been used to screen anti-cancer drugs. However, these experiments must be carefully designed to distinguish anti-tumor immunity from anti-HLA responses. If tumor and PBMC are from the same patients, human T cells are tolerated to autologous HLA on tumor cells. Human T cells can kill tumor cells only through recognition of tumor peptides presented by autologous MHC. This is the true anti-tumor immunity, and the response is usually weak. If PBMC from a foreign donor, then the human T cells can kill tumor cells by 2 different mechanisms. The first is the true anti-tumor immunity, as we just described, and the second is by recognition of mismatched HLA on tumor cells. This reaction is usually very strong and can control tumor growth, but this response does not occur in human cancer patients. And don't forget, as I described in the previous slide, most of the T cells in PBMC mouse model are activated by mouse MHC, and that can cause GVHD. A solution for this problem is using mice depletion of mouse MHC. These mice are now available from the Jackson Laboratory in US and Transgenic Mouse Model Core Facility in National Taiwan University. With this, I would like to conclude my talk and thank you for your attention.